So hi everybody, in this tutorial I will show to you how to use bigSCale step by step to cluster cell types and extract hierarchical markers in single cell datasets. One thing you need to know is that the current release of bigSCale is extremely user-friendly, meaning that you can fully analyze a single cell dataset with just one click of the keyboard. And in this sense, bigSCale is particularly useful and suited for the not so experienced bioinformaticians or even biologists who still would like to be able to analyze single cell datasets at a competitive level. So before starting, you need to know that for the moment bigSCale runs through MATLAB, so you must have MATLAB installed on your computer to be able to use bigSCale. And there are plans to release a standalone version in the future, but for now you need MATLAB. So let's start. Let's go to the GitHub repository page of bigSCale. You can see that there are two zip files. One is for Windows, the other is for Linux and Mac OS. This one has not been tested yet. It will be done shortly, but once it's tested it will run exactly like the one of Windows, so you can use this very same tutorial to know how to do the things. So basically we just download here the zip file of the bigSCale, and we unpack it. This I have already unpacked it. It's here in my desktop, the folder. So these are all the files which are needed to run bigSCale, but for you only these two folders are important. The input folder is the folder where you need to put your input files, namely is the expression counts and gene names. And the results folder is an empty folder where bigSCale will write the results of the analysis. So in the input folder I put a sample dataset. This sample dataset is the Linnarsson dataset published a couple of years ago of 3005 single cells from adult mouse cortex. And so you can run bigSCale and test that everything is alright by analyzing this dataset, and then you can replace these files with your own dataset. So as I said, there are two files. One is the expression. The expression counts is just the read or UMI counts for every cell and from every gene. And each row is a gene and each column is a cell. You just have numbers here, no gene names or cell names. And then the other file, it's called the gene names, and it has the same number of elements as the number of rows here in the expression file, plus it has a header. Please remember that you have to put a header here, because then bigSCale will use this header in the output files to write the header of the output files. So that's it. Basically, once you have the expression counts and the gene names in the input folder, you can start MATLAB and run bigSCale, which will do all the analysis for you. So here we are in the MATLAB main page. Here first we browse for the folder. So we have to locate the bigSCale folder in our computer. In our case it's here on the desktop, so I just go inside the folder and I press select folder. Once I'm there, I just press bigSCale brackets, enter. So it will just ask you one question in the beginning. It will ask you whether your dataset is made of UMIs or reads. And in our case, Linnarsson dataset is UMI based, so I press 2, enter. And then bigSCale will do all the analysis. A lot of text for the moment will keep flowing. Do not bother looking what's written. As long as it's flowing, it means everything is alright. So once bigSCale terminates the analysis, it states terminated. You can browse the results in the results folder. So let's go here in the results folder, which was previously empty. It's now filled with files, as you can see. Most of these files are for the more advanced usage of bigSCale, which we will cover in the next tutorial. Specifically, we will be explaining in this next tutorial how to how to tweak the parameters to adjust the output to your needs, how to remove batch effects, and also how to create iCells to analyze very large data sets of millions of cells. For the basic usage of bigSCale, we will be looking to this picture first, which is the dendrogram of the final clustering. This dendrogram is the same type of the dendrograms shown in the bigSCale paper, so I will not repeat how it's calculated. Please give a look to the paper. Here, for this dataset, the bigSCale found 5 clusters with 5 different colors. If you want to access the data of this dendrogram, you can use these files here: dendro order and clustering. Dendro order contains the ordering of the cells here in the same way of this dendrogram. You can see these are the columns, the order of the columns to achieve the same order here of the dendrogram. And in the other file called clustering, you have instead the cluster assignment. So for every cell you have a number which goes from 1 to 5 in our case, because we have 5 clusters, and it represents the cluster assignment, as you can see from the colors here of the dendrogram. So basically, with these two things, dendro, order, and clustering, with 2 files, you can have the data, the numbers that you need to make your own plots and things following the clustering of bigSCale. However, the most important output written by bigSCale is the one called is the Excel file called matrix bool. In this file, all the hierarchical markers of bigSCale are saved and structured by level. So let's go and give a look. Here below we have the different levels of the markers from level 1 to level 4. I will not explain what levels are. Please give a look to the bioRxiv paper again. From level 5 onwards, the sheets are empty because only the levels up to the number of clusters -1 are saved by default. So here what you have is the different markers, MVP, for instance, in MAL and PLP1 we have markers of oligodendrocytes, and you also have basically in each cell a score, which you have to keep in mind it's not an expression, it's a statistical score, it's a Z-score, and basically the higher the number, the more significantly expressed a gene is in a specific cluster. So one thing you need to know is that there are 2 ways to sort these markers, a pattern and a rank way. By default you will have all sorted by pattern, which means you can see more clearly here from level 3, which means that the pattern groups together the markers expressed in the same cluster. So for instance, you see here you have the first block of markers which are together because they are expressed from C1 to cluster 1 to cluster 3, although they are more strongly expressed in cluster 3. So, and then when you scroll down you will see other patterns. For instance, yeah, that's another pattern, but let's look to a more different one. Okay, so here for instance we have genes expressed instead in cluster 2 and then 4 and 5. So that's the first way to sort the markers. The pattern column. The second way is the rank column, which instead just simply sorts the markers from the strongest to the weakest one, where it is useful to know which one are the main drivers shaping the heterogeneity of your cells. So for instance, now we go to level 1 markers. Level 1, I already ordered them, you see, not by pattern but by rank. Well, if you don't know how to order in Excel, you just have to select all data and then press sort by rank, and then largest to smallest, and that's it. So we switch from pattern to rank. So let's go back, for instance, to level 1, and then the top scoring drivers are this one, HDR3A and GAD1, which are markers of interneurons, and then following you have all the other markers. I suggest to have a look first to the main drivers, so the genes, which will help you to identify what type, the phenotype of the cells, and then to dive more into the rest of the genes and heterogeneity by looking to higher levels and maybe look into this pattern sort instead of rank. One last thing, let's say that you would like to inspect the cell phenotypes in greater detail, for which I mean having more clusters than 5 here. For also in the case of Linarson, we know that at least 9 or 12 main cell types are present, so it would be better to increase the number of clusters here. So basically by default the bigSCale cuts the dendrogram at 20% of the total 3.8. So you see this distance here basically is the 20% of the total 3.8. So if you want to have more clusters, you should cut at the lower value. So instead of 20%, for instance, 15% or even 10%. How to do this is very simple. We just go back to the MATLAB page as before, and we type again exactly the same as before. So we type bigSCale and we press enter. Now bigSCale is aware of the fact that you are resuming the analysis to cut the dendrogram at a different depth, because he knows that there are files here in the folder. So by the presence of these files tells him that you want to resume the previously done analysis to cut the dendrogram at a different depth. So as I said, 0.2, which is 20%, would be the default value, but we want to have more clusters. So for instance, we press 0.1 and then enter, and then it will do exactly the same as before. And once it's terminated, it will say you can browse the results in the result folder, and you will have a new clustering and a new matrix with the new clusters and markers found. So thank you for watching this tutorial. If you have any questions, you can use the Google group of bigSCale, and the link of which you can see in the description of the video, or also in the GitHub README file. So once again, thank you to everybody, and stay tuned, and hopefully see you soon. Bye.